Good morning to everyone. So today we will study about the advantages, disadvantages, mechanism of CTDS system and the difference between the sustained and controlled drug delivery system. Okay. In last class we have studied definitions and that graph. So which is basic principle of CTDS system. Okay. So what is the advantages? The first one is total dose is low. Okay. So when we are comparing to a conventional dosage form, so here it is very low dose. Reduced GI side effect. Reduced dosing frequency. Dosing frequency means that multiple dosage regimen will be not there. So like how we are taking in a one day, we are taking 2 to 3 dose, right? So in CDDA system, that dosing frequency is not there. For one day, we can take one tablet, okay? Or for days, we can take one tablet. So like that. Reduced dosing frequency. Better patient acceptance and compliance. Less fluctuation at plasma drug level. So we can reduce that peak and valley in a CDDS system. More uniform drug effect we can observe in a CDDS system, and improve safety ratio also high in a CDDS system. So these things we can write when they may ask for comparison between the conventional dosage form and CDDS system. We can write this point also. Okay. So by having all these advantages, some of the disadvantages also there. So in a rare case it will be happening. So that is dose dumping. So dose dumping is nothing but the release of drug will be occurring in a sudden cases. Okay, so nothing but so dumping. So we are accumulating some of the drug in a particular site in a sudden cases. So that is called as dose dumping. Okay, so what happens when we are formulating a CDDS tablet, we are formulating such like that for 8 hours. So the drug should release its action for a particular time interval. For 1 hour this much, second hour this much, third hour this much, like that we are predicted and we are formulated. But what happens due to incompatibility of drug or due to instability of drug, what happens, the drug will release its action within 1 hour in a particular site itself. Okay, so due to that dumping will be occurring. Nothing but all the drug will be released at particular site within 1 hour or within 30 minutes. Okay, so that is called as dose dumping. Next, reduced potential for accurate dose adjustment. Okay, so in when you are preparing parenteral or nasal drug delivery system, and also in ophthalmic preparation when you are preparing, we are using this dose adjustment. So what happens in some cases, in a rare cases, it is reduced. The accuracy potential will be reduced. Need of additional patient education. So we have to educate the patient. So when we are giving a conventional dosage form, so just they have to administer morning and night, so how dosing frequency will be there, right? And they are habituated for that condition. So we have to educate them like, so no need to take that type of dosing frequency morning night, morning afternoon night, like that. So if you are administrating once means it will release its action, it will shows its action up to 1 day or 2 day. Okay, so like that we have to educate them before giving this serious tablet or capsule. So or else what happens, they will take that tablet like so how they were using conventional dosage form. Okay, so we have to educate them. Next, stability problem. Okay, so some of the drug may undergo physiological changes due to instability problem. So next, mechanism aspect over controlled drug delivery system. Okay, so many mechanism will be there, many approaches, so we can call it as approaches also. Okay, so different approaches will be there. So like rate pre-programmed drug delivery system, okay, automated drug delivery system, feedback regulatory drug delivery system, so many will be there. Okay, so in your syllabus only these three they have mentioned. Okay, and this is also dissolution, diffusion and ion exchange resin system. It will comes under rate pre-programmed drug delivery system. Okay, so nothing but the rate, the how drug should release, we have already programmed. Okay, so like that dissolution, diffusion and ion exchange resin system will be working. Okay, So first one is dissolution controlled drug delivery system. Okay, the main mechanism is dissolution. Okay, so by keeping this dissolution mechanism, we are pre-programmed this system. Okay, so in that dissolution system, we are again having two types, that is matrix dissolution controlled drug delivery system and encapsulation dissolution controlled drug delivery system. Okay, and the second thing is diffusion. Here the mechanism is diffusion. Okay, by keeping diffusion mechanism, how we can pre-program the rate of drug release. Okay, so in that also two types will be there, matrix and reservoir diffusion control drug delivery system. And the third thing is ion exchange resin system. Okay, so here we are maintaining the mechanism that ion exchange resin. Okay, in PIC or in analysis we have studied this ion exchange resin chromatography also there, right? So we are making a compatibility with ions and resin so that we can pre-program the rate of drug release. Okay, so these are the three main approaches or mechanism of controlled drug delivery system. Okay, so next is difference between sustained and controlled drug delivery system. Okay, so they may ask for 2 marks. What is the difference between sustained and CDTS system? The first one is under sustained, it constitutes dosage form that provide medication over extended period of time. Okay, so just it will give its action for a extended, nothing but prolonged period of time. Okay, so here also same, it constitutes dosage form that maintain constant drug level in blood or tissue. It will also gives its action for a prolonged time, but here we are maintaining the constant drug level. Okay, that's the difference. In sustained we cannot maintain the constant plasma drug level. So next is sustained drug release generally do not attain zero order release kinetics, but here in a controlled, so it will achieve zero order kinetics. Okay, the third thing is it usually do not contain mechanism to promote localization of the drug at active site. Here the CDTS forms contain method to promote localization of drug at active site. So nothing but in a sustained we are not adopting or we are not having any system to localize its action to a particular site. So nothing but targeting we are not doing in a sustained. Okay, just we are for example for GIT system we are taking sustained release means it will release its action over the GI system. Okay, and new method we are not following or new devices or new system we are not following in a sustained for a localization to an active site. But here in a control we can adopt or we can involve some of the new system to target its action. Okay, for example for what intestine only we had to achieve its action means we can target to a intestine, or we had to target to a liver means we can target to liver itself. So like that we can adopt or we can involve new system or new devices to achieve localization to active site. So these are the some differences between sustained and controlled. It will come for 2 marks almost. Okay, so next class we will discuss about the that approaches. Okay, dissolution, diffusion and ion exchange resin. Okay, thank you.